sitting there playing like, I don't know, some video game. And he's like, yes, yes. And then my favorite one is like, it's 5 o'clock and one person, looks at his watch. He's like, oh, I'm so sorry. I have to go to another meeting. And he's like going like, happy hour or something like that.
Oh,
and
right.
So they really seem to kind of have a sense of what we'd be facing, this last year.
Mm-hmm. Was there anyone in this example doing some like arts and crafts beneath the table? I think that's a good one.
Yeah, I think so. Someone had the dog. You've always have someone with like the really loud dog. I don't think they had anyone who had sort of like the child kind of like wander in like that CNN interview. So so yes, it's yes, very 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 interesting. But I guess we're all hoping that by fall we should be back to normal. I guess the hope for meetings and stuff like that.
Yeah, fingers crossed. It's hard to know how much it's going to be an immediate versus sort of a slow climb, in terms of events and event size capacities and things like that. So we'll see. But it seems like we'll slowly get there.
Yeah, I really wonder in terms of how much, right, they'll limit meeting size, in terms of, they say even when people are vaccinated, they're going to want to have some types of distancing and mask use. So I really do wonder. But yeah, it was, yeah, I feel bad for my, friends in other institutions who are actually are teaching and have to teach with a mask on, for like, it's like, I find that, it's after a little bit wearing a mask, it gets a bit uncomfortable. But I guess I know they adjust to it. But
sure. Yeah, a good friend of mine is a middle school science teacher, and she's done a pretty exhaustive testing of various masks because she needs to talk in hers all day. And so I think she sort of slowly evaluated and found the best mask, and she has one for the morning and then she switches it out at lunch and then she washes them weekly. She's got a whole routine, but it's a real challenge. It's even different than just being able to walk quietly down the street. When you're really having to talk into it, that's a different beast.
Yeah, I'm grateful. I mean, I thought like in Boston, if you were out doing anything, even running, that you had to have a mask in public, or maybe I misconstrued that. But I mean, so that's why I'm lucky. I run really early in the morning, and obviously in the winter, you have like a balaclava or something over your face. But
right.
I couldn't imagine like in the summer, having to run with a mask on. That would be painful.
Mm-hmm. I know. I've come to sort of appreciate it during the winter because it keeps my face warm. But during the summer is pretty brutal. Okay, great. We've got a good number of attendees here now, and it's 1:01. Yeah. So I'm sure a few people will continue rolling in, but I will go ahead and get started by welcoming everyone. Thank you so much for coming. Welcome to a new year and a new year of MSI seminars. Today, to kick off, I'm really pleased to introduce Dr. Joel Freundlich, who is an associate professor at Rutgers University in the Department of Pharmacology, Physiology, and Neuroscience, as well as the Department of Medicine. His undergraduate and master's degree training were in chemical engineering at Cornell as a McMullins Dean Scholar. He received a doctorate in organic chemistry from MIT here in the Boston area under the tutelage of 2005 Nobel Prize in Chemistry awardee Richard Schrock. Dr. Freundlich spent 8 years in the pharmaceutical industry as a medicinal chemist and then returned to academia with stints at Princeton and Texas A&M. He's been at Rutgers since 2011, from what I can tell. And there, the Freundlich Laboratory is positioned at the intersection of computation, chemistry, and biology. The laboratory works to develop new tools that contribute to our understanding of the pathogenesis of infectious diseases, especially those caused by Mycobacterium tuberculosis and the ESKAPE bacteria, and translate these findings towards the discovery of novel therapeutic strategies. So as regular MSI seminar attendees will notice, we are in a slightly new format here in the Zoom webinar so that we can have a little bit more interaction in real time. And also that will allow during our Q&A session following Dr. Freundlich's talk for you to raise your hand, and we'd be happy to unmute you so you can ask a question verbally. Also feel free, if you have a question that you don't want to forget while we're going along, feel free to put that in the Q&A section, which you'll find that icon at the bottom of the Zoom webinar screen. So without further ado, I will turn it over to you.
Great. Thank you so much, Hannah. It's really a pleasure to be here today. I appreciate the kind introduction and invitation to visit with everyone. I'm sorry that could not be visiting in person. Everyone's staying safe and well. And let's just see. So it should just take a second for PowerPoint to convince itself. Second. Great. Can everyone hear me okay?
I can, yeah. Pause for a second there, but you're looking good now and I can see your screen.
Okay, excellent, good. Okay, and let's just see if I can move this over a bit. Okay, great. So I'll start and I'll try to keep my timer on to watch our time a bit. Okay, perfect. Well, really a pleasure to be here today. And what I'd like to do today is talk to you about how the lab has tried to balance computation, chemistry, and biology to come up with new platforms for infectious disease research. Okay, great. And certainly in this seminar series, and given the current climate, right, this slide almost is, not needed, which tells us a lot, probably how far we've come in the last, what's transpired in the last year. Even before SARS-CoV-2, we could see that infectious diseases were amongst the leading causes of death worldwide. If we look at tuberculosis, we can see that really every year, and I didn't go back, but at least as long as I've been in the field, for about 10, 15 years, you've been looking at, 10 million cases a year, unfortunately leading to about 1.5 million fatalities. Driver of infectious disease impact of global health. Certainly in the US alone, if we take that sort of a narrow view, drug resistance is a major issue, both in terms of number of infections and in deaths. And obviously I just had to update this number today, which is, sad. And, but we're approaching about, at least the stats I could see from the Hopkins website, about 2 million deaths from SARS-CoV-2 as of today. So we have a very significant issue here with infectious diseases and global health. If we look at tuberculosis as an example, we can see that we have drugs available. We have frontline drugs and we have second-line drugs. But with each of them, we have established clinical resistance, which among other issues, such as the need to reduce the length of treatment, really necessitates the need for new therapeutic regimens. And so for this, as we've thought about this, it's really brought up for us and others grand challenges in infectious disease research. One, I think, is just a need to continue to better understand the basic biology of the disease state. And then two, to leverage that basic understanding to help discover and then optimize novel therapeutic regimens. And by therapeutic regimens, I mean both molecule, the drug potentially, or the beginnings of that, and the molecular target. And so when we came into sort of the TB world about 13 years ago, we were cognizant of papers that really helped shape the field in TB, but then also in antibacterials in general. And this paper here by David Pompliano and colleagues when they were at GSK, I really believe this had a large impact on the field. At the time, they summarized a 7-year effort at GSK, taking a number of genes that they believed to be essential for infection for both gram-positive and or gram-negative bacteria. They conducted 70 high-throughput screens where they looked at essentially small molecule libraries being assayed for their ability to inhibit the function of that gene. And what you could see is the result was really disappointing. Really only 16 screens gave hits, the beginnings of early molecules, perhaps showing in vitro potency. And only a few of them were able to be optimized, most likely to compounds with whole cell activity, perhaps some early beginnings of in vivo activity in a relevant animal model of infection. But overall, 7 years and more than $7 million gave them very little when they focused on targets. And this and other contributions to the field, I believe, really led to a shift in approach to more of a concern and a focus on compounds that inhibit the growth of the bacterium in culture. And you saw basically high throughput whole cell phenotypic screens, looking at up to millions of compounds being assayed for growth inhibition, and then trying to worry about how the molecules act in terms of mechanism of action later. And so it really led to a downgrading of target-based approaches. And so when we came into the area, we and others obviously were really thinking about this, the current background or the current sort of setting, and it led to some basic questions for us. One was this very basic question is whether target-based approaches still can be viable. Secondly, if we embrace the high throughput whole cell phenotypic screening approach, we think that we know that we're generating a lot of data, and we're really in this realm, this sort of buzzword, big data, hundreds and thousands of compounds being assayed against a bacterium, and hopefully in the best of cases, that data being publicly available. And we ask the question, with machine learning approaches, how can we leverage this information to not only discover novel antibacterials, but also help evolve their properties? And over the course of time, we basically looked at these questions and tried to pursue answers from our approach. And our approach basically melds different ways of finding molecules: computation, experimental screening, structure-based design, to arrive at novel antibacterials, and then to understand their target. And the deliverables thus far, and I'll talk about these today, have been computational models. They've been chemical tools to probe basic biology. We've been fortunate to see preclinical drug candidates, 2 for Neisseria gonorrhea that I won't talk about today, and then 1 for tuberculosis. In exploring these molecules, we've been able to validate novel drug targets and also to put forth novel biological tools. And so let's begin with this first question about, can target-based approaches still be viable? I'm going to take you through a story that begins with the use of, or really leveraging a high throughput screen from GlaxoSmithKline that led to them to put out to the community, very nicely so, 168 compounds that had demonstrated whole cell activity against TB, but for most, if not all of them, they were basically devoid of target information. And we, in collaboration with David Aland's lab, used a reporter-based assay, this INIVAC screen, which, in David's experience and ours with him in collaboration, has basically picks out compounds that are engaging the cell wall in Mycobacterium tuberculosis. And so this finds compounds, drugs like isoniazid and ethambutol, and it lit up for this compound DG167, this indazole sulfonamide. And this is a project that was led in our lab by Daigo Inuyama, who was a postdoc, who's now at Schrodinger, and Divya Awasthi, who's now at Seattle Genetics. Pradeep Kumar did a lot of the microbiology in David Aland's lab, and Glenn Capodagle, led the structural biology in Matt Nedge's lab. And this work has been published in two papers, most recently one in 2020. So this was an interesting compound for us because it exhibited sub-micromolar whole cell activity. So for all of you, I know it's probably commonplace. So the MIC, the minimum inhibitory concentration, for us, it's what the minimum concentration that gives you 90% growth inhibition in culture. So that's good. It had a lack of relative cytotoxicity to Vera cells, which are a common model mammalian cell line. You obviously want to inhibit the growth of the bug without inhibiting the growth of the mammalian cell line. The compound had good solubility. We like this to be above 100 micromolar, but it had poor metabolic stability. This is a mouse liver microsome stability. It's an in vitro assay using the membrane-bound fraction of mouse hepatocytes. It typically gives you phase 1 metabolic events. And you'd like that to be above an hour. And this was woefully small at 10 minutes. We were able to tell that the primary metabolic event was removal of this methyl group to give you this parent indazole that we synthesized, and it was whole cell inactive. So you have an inactivating metabolism. And this metabolic instability leads to suboptimal PK. This is in CD1 mice performed by Véronique Dartois' lab in collaboration. You're looking at 3 mice, 25 mg per kg, 1 dose oral, or a single 5 mg per kg IV dose. And in general, the exposure, although not terrible, is not great. And this in turn manifested itself in the lack of in vivo activity in this mouse model of subacute infection. So you have infection, you let that go for 2 weeks, and then you dose once daily oral for 4 weeks. I'm showing you 2-week data. The 4-week data was roughly the same, but DG167 essentially leads to a growth of bacteria in the lungs of the dosed mice compared to the inoculum. So there's no activity, there's no reduction in infection like you see with isoniazid as a positive control at 25 mg per kg. So we wanted to improve this molecule, and we really wanted to think of a target-based approach. And we were fortunate that Pradeep could raise spontaneous resistant mutants, roughly with a frequency of resistance of 10 to the -8 in the wild-type TB background of H37RV. The mutations basically were all in Cas-A. This is a beta-keto-acyl synthase, condensing an acyl ACP with a malonyl ACP to give you the beta-keto-acyl ACP. This is a fast 2 cycle of fatty acid biosynthesis in TB. It ultimately gives long-chain alpha-alkyl beta-hydroxy fatty acids, mycolic acids, that are a key component of a cell wall. And there are other beta-keto acyl synthetases within TB. Two of them believed to be the only other two are Cas-B and this initiator Cas beta-keto acyl synthase. These are not essential in vitro. Cas-A has been found to be essential in vitro. Compounds have been reported in the literature to modulate these beta-keto acyl synthases in TB, thiolactomycin or potensomycin. However, they didn't move forward to key in vivo studies of efficacy because of a lack of whole cell activity and acceptable mouse PK. We're fortunate that knowing that we were targeting Cas-A, Matt Nitich's lab solved the crystal structure of Cas-A with DG167 bound. In one of the monomeric units of protein, you see, interestingly, two molecules of DG167 are bound with a binding affinity on the order of about 100 nanomolar. So you're showing that you are engaging the purified protein. You also are engaging this pathway by looking at this fatty acid TLC. So you essentially can look at how the small molecule inhibits basically the formation of mycolic acids. That's in pink, the end product that goes down in a dose-dependent fashion. And the short chain fatty acid precursor goes up because you're stopping that critical elongation. Similar to the frontline drug isoniazid, which inhibits INHA, which is also in the pathway. The whole cell inactive compound does not have this phenotype. If we perform this assay with a spontaneous resistant mutant to DG167, you again see loss of this phenotype. So you are engaging Cas-A in the cell. So with this information, with target information, Daigo came up with a structure-based hypothesis that we essentially could move this nitrogen that's giving us the lability of this bond to the methyl group over to this position in the ring and give either another indazole, two nitrogens, or an indole, this is a carbon, and we could potentially have better compounds. And he led an effort that was roughly about 300 compounds worth of chemistry. And essentially what we ended up with, and hopefully I'm not obscuring it with my little control, we ended up with the best compound being JSF 3285. It is an indazole. We've moved this nitrogen over here. We've added a 4-fluorobutyl group on the sulfonamide. And what we see is a progression of properties when we look through molecules in this optimization. With 3285, we have better potency, better solubility, better mouse liver microsome stability, and most importantly, 10-fold better PK. This is over a 24-hour window. We're getting better drug exposure in the plasma and also in the lungs of mice. The compound maintains nanomolar binding affinity for Cas-A. In a crystal structure, it also binds similarly to one of the molecules of an inhibitor in the cell. In the unit cell, the protein. There is only one molecule 3285 bound in the active site as compared to two with DG167. It inhibits fatty acid biosynthesis in the cell. And critically, when we go to the subacute model again, where when we look at dosing after 14 days of infection, whereas DG167, our beginning gave growth, and then another compound that came after it gave growth, another compound, an indole, gave static behavior. Finally, at 100 or 200 mg per kg once daily oral, we get reduction in infection. And that was a really gratifying result. We then move on to the more important chronic model where we've we've infected, let that go for 4 weeks. So we're getting much more established infection. And we can see that the minimum effective dose for our compound 3285 is 5 mg per kg once daily oral. And that really is in the dosing regime of frontline drugs. Isoniazid at 10 mg per kg or rifampicin at 10 mg per kg are very similar in efficacy to 3285 at 20 mg per kg. If you add 3285 to either of these frontline drugs, you get a slight improvement in activity over the frontline drug alone. And most importantly, because TB needs to be treated in combination clinically because of resistance and the need to really try to reduce the time of treatment, you can see if we look at the frontline therapy of isoniazid, rifampicin, pyrazinamide, and ethambutol, HRZE, we can see if we replace isoniazid with 3285, we get a 0.9 log improvement in the ability to reduce infection in lungs. So this importantly is a validation of a preclinical drug candidate. And this provides rigorous in vivo validation of Cas-A as a TB drug target. And so at the moment, we're currently taking the compound on to critical animal safety studies and doing further drug combination studies. So hopefully with this short story, I've had a chance to show you that indeed, one with the right tools and obviously good fortune, and being thorough can make target-based approaches still work in this day and age. So we then move sort of to the other sort of approach, and I think what's been much more commonplace, the screening approach. And this idea of can we leverage all this data where we're so resource limited that really we're only able to take less than 1% of our best compounds in a screen, the hits, and try to move ahead with them. And so we kind of joke that it's sort of like the needle in the haystack. And can we use computation to learn from those results? And so we began this back around 2010. And at the time we were collaborating with Sean Eakin's lab and his company, and we were interested in using Bayesian modeling, conditional probability, to describe the results from high throughput screens, to characterize the hits and the inactives. And at the time, I had a really simple-minded thought. I just thought, well, we're building these predictive models. Can we use them to predict new compounds? After all, that's what we're after. We're after finding new antibacterials. And so really these models offer a way to learn from the past. That past is screening data that's publicly available. Hundreds of thousands of compounds often can be available in PubChem, Chembl. So they're available for all of us to look at and to utilize. And so this is a binary classifier model. You as a user set the classification or the threshold for activity. So for example, we would say for TB or a lot of other bacteria, an MIC at or below 10 micrograms per mil or 10 micromolar, depending on the units of your screen, would be active. And an MIC above that would be inactive. So good, bad, you separate them. And then the nice thing is that once you've built a model and you've statistically validated it, you can score a new library of compounds that aren't in the training set. And the score for each compound scales with the probability of that compound meeting or exceeding your activity threshold that you use for the model. And so when we began, we worked with Accelerus, which became Biovia, Discovery Studio, or Pipeline Pilot. And we wanted software that everyone could use. And we used a very simple set of descriptors. This is not a large set like we would use today, but basically descriptors that describe the physical properties of the molecules, and then a standard way of describing molecular structure, fingerprints, in this case, FCFP6. And so essentially the model correlates predicted properties and structure with activity or inactivity. It just is very quickly. You can build the model and score and statistically validate it and score a million compounds within a few hours, up to maybe about 12 hours. So it's very fast. We can then we can validate our models with standard procedures, typical k-fold cross-validation, where you're basically leaving out part of the training set, rebuilding the model with basically what remains, and then predicting what you left out. And typically, we like our statistics that tell us about how well it finds actives and inactives and is correct overall to be at least above 70%. We're not after necessarily the best models here. We are aware of the issues of overfitting, but also we just want models at this point that are practical. And we are coming back to this in terms of the finer details of the modeling, just to look at how we approach optimal model performance. And so we take these models, we score a library of compounds that are commercially available that, to the best of our knowledge, have not been assayed en masse against a bacteria for growth inhibition, for example. We take the top scoring compounds, and this is where we extend beyond. At the time, there was a single report from NITD, the Novartis Institute for Tropical Diseases, in 2008, where they built a model, but they didn't report any predictions and any testing of those predictions. So we predict compounds, and then we assay them in our lab to find out how the models perform. And so, for example, in a model knowing about how compounds inhibit the growth of drug-resistant staph, that I'll come back to later, we could see that we could take this model from about 1,600 compounds. We can score a 3 million compound library from enamine. We can do this on the order of hours. And then we can take the top scoring compounds, in this case, we took the top 15. 3 out of those 15 had MICs in our hands experimentally at or below 10 micrograms per mil. So there's an enrichment in actives. And 8 out of the 8 bottom scoring compounds, so the least likely to be active, all had MICs at or above 50 micrograms per mil. So the models are generally very good at triaging inactives. And you can do this on the order of days. You don't need a high throughput screening to do this experimentally. You don't have to purchase all the compounds. And so we really believe this is very time and cost efficient. And we believe it's a discovery engine for novel antibacterials. And I've shown here some of the successes of the models. There's certainly your failures always, but some of the successes with a number of different bacteria. And I'll talk first about results with Mycobacterium tuberculosis. This is a project that was led by Xin Wang, who I believe is on the meeting today. He was a graduate student. He's now a postdoc with Eric Rubin and Sarah Fortune at Harvard. And again, Daigo Inuyama, who was a postdoc who's now at Schrodinger. And so we were interested in this triazine that we published in 2013, and then a follow-up paper that Xin and Daigo led in 2020, because of the nitrofuran. In many therapeutic areas, you would say that that's sort of a no-no. There's concerns about mutagenicity. This compound is actually not mutagenic, Ames mutagenic. But when you look at recent clinical additions to the TB drugs, we see two nitroimidazoles. Pretomanid, that was approved in 2019 as part of a 3-drug therapy for MDR or XDR TB, or delamanid, which is approved in the European Union. So these are important additions to anti-tubercular drugs, but they're not perfect, and they have liabilities that are driving the approaches and efforts towards second-generation drugs. They have rather high frequencies of resistance, 10 to the -5, 10 to the -6, that lead to very significant losses of activity, up to 1,000-fold shift in MIC. Delamanid also has an issue with poor oral bioavailability, I'll say that twice. And so there's really a need for new compounds, and we wanted to explore this triazine further. Daigo led a lot of chemistry. I'm just showing one slide. Here you can see that he looked at, from our knowledge of the SAR, the structure activity relationships with pretomanid and delamanid, that the nitro group is important. Indeed, when he replaced it with other groups, you lost activity. And a lot of chemistry we do in the lab, but it's always meant to arrive at compounds quickly and easily so we can explore biological activity. Xin jumped into looking at the mechanism of action and began with spontaneous resistant mutants to 2019 raised in the wild type H37RB background. Right away, we saw a difference with pretomanid. We saw a much lower frequency of resistance, low 10 to the -6 to high 10 to the -8. This compares to pretomanid at 10 to the -5, 10 to the -6. So already seeing a difference. Also, when we look at 2019, in the 17 resistant isolates that Xin worked with, he saw a 16 to 64-fold loss of activity, whereas pretomanid, which is cross-resistant, gave us some strains with a 64 to 512x loss, but many with greater than 1,000-fold loss of activity. So this is what we said, that pretomanid and delamanid lose a lot of activity against these types of resistant strains. These strains, upon whole genome sequencing, harbored mutations that were familiar to us from the literature. And specifically, a lot of that literature is thanks to the lab of Cliff Berry at NIH. These mutations occur in the biosynthesis of this diazoflavin F420, FBIB, FBIC, and also in its reduction with the F420 dependent glucose phosphate dehydrogenase, FGD1. These can be used, as Cliff has shown, to power a nitroreductase, in this case DDN. And this actually leads to the reduction of pretomanid to release NO and generate what's called this des-nitro pretomanid. And so naturally, with knowing that these mutations and the literature from pretomanid, we wanted to see if 2019 functions similarly. And to do this, Xin wanted to explore mechanism by looking at the fate of 2019 within TB. So this idea of looking at intrabacterial drug metabolism came up, sort of turning metabolomics on its head. Instead of looking at what the drug does to the bug, let's look at what the bug does to the drug. And so Xin really stood on the shoulders of giants such as Josh Rabinowitz and David Botstein, and then Q Ree, who took this methodology to TB and also worked from Veronique Dartois' lab. And so he used basically a liquid culture approach where you're taking aliquots, you're basically isolating the cell pellet, washing that, quenching metabolism, a la Rabinowitz and Botstein, with cold acetonitrile methanol water, and then further processing the sample till you have the cell lysate, and you can interrogate it by liquid chromatography mass spec. And so what Xin did was essentially begin by looking in the cell to see if there's accumulation of the parent compound 2019. If you look in the wild type background, you can see over time, there's basically an accumulation of 2019 inside the bacterium. And then you may recall that we saw mutations in FGD1, so we thought it would be important for metabolism. And indeed, if you inactivate that with a transposon insertion, you see basically increased accumulation of 2019 inside the cell. And then you want to look in the cell to see basically what evidence you can find for metabolites. And to date, we've been able to definitively identify one metabolite. And this is an analogy to the pretomanid biotransformation. We see the loss of the nitro group. So give us what we call the des nitro compound, or 2026. And in the wild type background, this basically accumulates over time. And if we inactivate F420 with this FGD1 transposon insertion, we see a failure of this to accumulate. So we've shown by IBDM there's a F420 dependent biotransformation. Shin went on to show that basically NO is indeed formed. We can use a Grice assay, and you're looking at ultimately nitrite formed and released into the media. Protominid shows significant release of nitrite, so does 2019. If we look at performing this instead of in the wild type background with the transposon mutagenized FGD1, we see no significant formation of NO. So again, we have an F420 dependent metabolism inside the cell to form the des nitro compound and release NO. And before we leave this, I really like to say that I believe, up until recent times, I think that the field, both in TB and outside of TB, really has failed to think of biotransformations within the bacterium as being really prevalent. I think we've always agreed, I think, and known that there are isolated examples due to wonderful work from investigators with isoniazid or pyrazinamide, for example, for TB. But we think that this is much more prevalent. And so just some examples from our lab: this methotrexate diester gets hydrolyzed inside TB. The steinopyrimidine with an ester does get hydrolyzed. Also, there's a lot of other chemistry this undergoes that we're trying to ready the publication of the manuscript. And then finally, this nitrobenzofurazan that Shin also worked on has a nitro group, but the metabolites we see are not similar to what we see with protominid or 2019. The nitro group does not get cleaved to release NO, and instead gets reduced to give you an amine. But then even more interesting, it gets reduced to give an amine, and this oxidiazol ring actually shifts along the benzene core. And that's, at least my eyes, a really cool biotransformation that we still don't quite understand. And so these examples are joined by others from Curie's lab, Carl Nathan's lab, Cliff Berry's lab. And we really think this is much more prevalent. We actually have a manuscript ready to submit in E. coli talking about these biotransformations. And so again, we really think this is a shift in how we can view the stability of compounds within bacteria and their effects on efficacy and mechanism. Shin wasn't satisfied with this level of understanding. He went on to show by RNA-seq that 2019 clusters with isoniazid and ethionamide, two drugs that basically inhibit INHA. So this is again coming back to the FAST2 cycle. He showed that the 2019, but not 2026, inhibits INHA, purified INHA. And then also within the cell, again, returning to the FAST2 TLC approach, he can show that in a dose-dependent fashion, you see a depletion of mycolic acid formation, the end product, and an accumulation of a short-chain fatty acid precursor. So demonstration that you are inhibiting FAST2 pathway. And if you look at overexpressing INHA with this strain from Bill Jacobs' lab, we lose this phenotype, suggesting that, again, the FAST2 inhibition is due to engagement of INHA in the cell. But curiously, if we repeat the experiment with the FGD1 transposon insertion, we actually also lose this phenotype. And so where we're left with is this current understanding of the mechanism of 2019. We have inhibition of the parent compound of INHA. We have an F420 dependent metabolism to release NO and afford this metabolite. But this metabolism also seems to be implicated in forming one or more metabolites that we've yet to identify that also has an effect on inhibiting the FAST2 pathway, if not INHA. This is a different mechanism from the clinical compounds, protominid and delaminid. They also extrude NO, but they have effects downstream in mycolic acid biosynthesis, specifically with affecting hydroxymycolate levels. And so we believe this is a differential mechanism for these nitroheterocycles, and we're hopeful that this will have a significant impact on the search for second generation clinical compounds. So with that work in TB, I want to stress that this discovery engine with machine learning can be used with other bacteria. We've done this with staph, and I'll quickly go through this. This is work from Jimmy Patel, who was a grad student, who's now a med student, and Alex Perriman, who was a postdoc, who's now at Repair Therapeutics. They built a model. They scored a 3 million compound library, and they found this quinoline, which has good activity against MRSA, as well as methicillin sensitive strain, and has a good cytotoxicity window. I think the motivation for exploring compounds active against drug-resistant staph is obvious because of the number of infections in the hospital, the rise of resistance, and the limited options therapeutically. Jimmy jumped from the chemistry into the biology. He showed the compound is cidal against MSSA and MRSA strains. Against MRSA, we get very quick sterilization, where basically after 4 hours at 1 XMIC, we sterilize the culture. Vancomycin, for example, in comparison, is is much slower. The compound has activity against vancomycin-resistant strains, clinical strains. So the model did what it was supposed to do. It found an active compound against a drug-resistant staph. The model did not make predictions for the compound's other properties, solubility, stability, and PK. Those are actually quite poor, and they manifest themselves in very poor behavior in a mouse MRSA peritonitis model. Even with a suboptimal formulation and a suboptimal dosing of compound, we still could see some survival, albeit very small, with 3151. So we believe there still is an opportunity to move ahead this compound once it has better properties that were not predicted with the model. Jimmy raised spontaneous resistant mutants with a frequency of 6 × 10⁻⁸. They 3 strains were sequenced. They had high levels of resistance and a mutation in the promoter region of this gene YCEI. Resistance has not been, to the best of our knowledge, published in staph. It has been shown in Burkholderia cynosopacia to encode a bacterial lipocalin that sequesters hydrophobic drugs. And so we were led to a hypothesis of mechanism of resistance that overexpression is leading to resistance. And indeed, Jimmy's been able to bear that out. This is a great collaboration with Jeff Boyd's lab, our staph genetics experts. Jimmy jumped right into this. He showed by qRT-PCR that indeed with these 3 mutant strains, we have overexpression of YCEI. He was able to take a plasmid, incorporate a single copy of either the wild type allele or the mutant allele into the wild type strain in the AppB site. And you can see that incorporation of a single copy of the wild type allele leads to resistance. We can inactivate with a transposon the wild type allele and repeat this type of experiment, and again see only when we bring in a single copy of the mutant allele do we get resistance. And then finally, Jimmy was able to overexpress YCEI with this PEPs5 plasmid with using xylose induction, and again get resistance. So these experiments, we believe, supports this idea that YCEI overexpression leads to resistance. This, to the best of our knowledge, when we can get this submitted and accepted, hopefully, will be the first reported example of this type of resistance within staph. And finally, before we leave this, we've been recently trying to look at the effect with resistance through mutation in YCEI with intrabacterial drug metabolism. We can see in the wild type strain, when we compare this to one of the mutants, we see a decreased accumulation of 3151 in the cell. So this decreased exposure of staph to the compound with this mutation. And we've been able to identify one metabolite. Again, we don't see loss, we don't see extrusion of NO. We instead see a mutation, a metabolite where we get formation of the amine. And again, this is reduced in the mutant. And then finally, we've been able to extend this methodology with machine learning to gram negatives. Alex Lemens, who was a grad student in Nancy Connell's lab, who's now a faculty at Rutgers, and Alex Perriman again, looked at this obligate intracellular gram negative relevant to biodefense. They are able to, Alex L was able to come up with a really nice screen with Vero cells infected with Rickettsia canadensis expressing GFP. He was able to screen a library of 2,000 compounds. And from Microsource, Alex P was able to build a machine learning model with this. And we took the top 82 scoring compounds. 5 out of the 82 had IC50s at or below 13 micrograms per mil. One, for example, is this indole amide. This is a novel chemical structural type for an antibacterial. And we're currently trying to finish off this manuscript to understand something about mechanism and then try to move this forward. And then with Neisseria gonorrhea, which obviously represents a very significant issue in terms of global health, in terms of the rising number of cases and drug resistance. Currently, standard of care is ceftriaxone and/or azithromycin. This is work led by Janina Cruz Pereira, who was a postdoc in the lab, who's now a data scientist at Vant AI, and Sam Dehaer, who's a current postdoc in the lab. They were able to build a model with a relatively small amount of data, 282 compounds from Kemble. They then built the model and validated it. They scored a library of compounds from Azinex, 190,000. And importantly, they took the top scoring 10,000 and they filtered those to select the 100 most diverse. So you're spreading your bet amongst different chemotypes. 2 out of those 20 showed activity at or below, with an MIC below 8 micrograms per mil. We have this pyridone, or this tetrahydrofuropyrimidine dione. These are novel chemical types for antibacterials. And we published this in 2020, and we're currently right now trying to understand mechanism as well as advance these compounds. So hopefully with these limited examples, I've been able to convince you that machine learning can leverage this big data that's out there available for us to use to come up with novel antibacterials against TB, Staph aureus, and then gram negatives, all of global health relevance. Others are certainly in this space. We welcome that. It's great to see others, most notably from the Boston area, Jim Collins' lab, Brian Bryson's lab have had wonderful contributions in the last year or two, and we look forward to further innovation in this field. So hopefully with this work, I've convinced you that we can embrace this high throughput screening environment, take the big data that's being generated and use it to build predictive machine learning models to predict novel antibacterial chemotypes. Well, as we saw in the CAS-A program, you can get a hit from a screen, whether it's computational or experimental, but it's most likely not going to be a drug. It's most likely not going to have all the properties, and it's going to need to be optimized. And so can we leverage big data to evolve properties? And I'm really just going to summarize this basically for the sake of time, but we've been able to build, validate, and publish models, machine learning models, Bayesian models for mammalian cell cytotoxicity, Vera cells, for metabolic stability, that being mouse liver microsome stability, for aqueous solubility. And then just actually yesterday, just accepted was a random forest model to predict oral exposure in the plasma of small molecules in a mouse model for PK. And actually, I just want to highlight this. This is actually work by Hasib Mughal, who's an undergraduate in my lab. He's applying to grad schools. Perhaps some of you may have seen his application, but he led that effort really pretty much on his own. And others have joined this space. There's multiple papers from pharma, but the issue with those contributions from pharma, no matter how wonderful they are, they often can't turn out their model to the community because it's built on their own compound decks. We can turn out our models and hope that people will use them. We're obviously joined in the space by amazing contributions from example, in the Boston area, Regina Barzolet's lab, Connor Coley's lab, both at MIT, and really think there's just a lot more to be done in this area. So hopefully, even in that short slide, I've been able to convince you that we can leverage big data to improve other properties of molecule besides in vitro activity to really help make our way to compounds of translational significance. And so in summary, in this very quick overview, we've obviously been focused on this global health dilemma that is represented by infectious diseases, and this presenting tremendous challenges for our field. And hopefully I've been able to convince you that with an approach combining computation, chemistry, and biology, we can contribute to trying to move towards novel therapeutic regimens. We can bring to the field computational models to find antibacterials and to evolve their properties. We can, obviously as an outgrowth, we can have molecules that can explore mechanism and help validate targets. We can hopefully be fortunate enough and thorough enough to come up with preclinical drug candidates. I told you about a TB compound, and also we have two compounds for Neisseria gonorrhea infection. And then finally, we can also bring to bear biological tools, such as looking at intrabacterial drug metabolism, which ultimately can help arrive at better compounds, but also help reflect on mechanism. And so finally, I'm really just the mouthpiece. I've just been so fortunate to have a fantastic group. I've highlighted the contributions of many people here today. Wonderful collaborators, NIH for funding, Dassault Systems for software, and you for your attention. I'm happy to answer any questions. Thank you.
Awesome. Thank you so much.
Thank you.
Great. Yeah, we have, now we're going to talk over each other. Sorry, Mike. I was just going to say that we have Mike Gilmore here in both capacities as an MSI co-director and also as the head of BARN, both super relevant to this talk, which we're happy to have many members of both of those communities here in attendance. So Mike's going to help moderate our Q&A discussion here. And feel free to either put Q&A in written in or typed in the Q&A feature, or to raise your hand, which I see a couple people are already doing. So Mike, take it away.
Great, thanks, Hannah. I can't see the raised hands, and so maybe you can.
Oh, I'd be happy to call on those. Yeah.
That would be great. I'll start. We've got a question first from. Well, first, let me say that was terrific, Joel. Thanks so much.
Thank you.
And,
Thank you.
We consider the Boston Area Antimicrobial Research Network, BARN, to extend all the way to New Jersey, to Brunswick, anyway. So that was great. Tom Stratton asked, did you consider synthesizing a dimer of DG167, perhaps at the labile in methyl group or JSF 3285, given that the two molecules were visualized in the active site of Cas-A?
Great. And if I think it's a Tom Stratton, then I know. So you're seeing this is a Tom Stratton who I for lack of time, I didn't get to his project. He actually was an undergraduate in the lab who did phenomenal things. He went on to do his PhD with Phil Barron at Scripps and is now a scientist at Gilead. So if that's the right Tom Stratton, so it's a really good question. And so we've looked at that. So the crystal structure, you may recall, showed two molecules of DG167 bound to Cas-A. And so we certainly were drawn to think about how we could connect those two molecules. We've been unable to do so in a productive fashion. We think right now just we're not getting the angles correct to basically guide ourselves or link from one molecule to the other. But the curious thing is that if we look at the potencies of binding, and so with microscale thermophoresis, Matt Nedich's lab was able to calculate, I believe for DG167, where you have two copies binding the protein, effectively it works out through the formalism to be calculated as EC50. That was, let's say, I think about 130 nanomolar, whereas the single molecule 3285 that we ended up with had a KD of 70 nanomolar. So it's really led us to think that, I'm not sure if that second molecule, the B molecule that we're not mimicking with our better compound, is more kind of brought along for the ride. I think it's making some contacts, but I'm not sure it's adding so much, at least with the interactions we're seeing right now, to binding affinity, and certainly not to whole cell activity. That being said, I think Tom is completely correct, in that this is a way to go forward to basically to further gain other interactions with the protein. So we have been unsuccessful to date yet, but I think Tom is completely correct that would be a great way to go forward.
Thank you. Let's see, we've got a question from Kim Lewis. I will allow you to talk. Maybe.
I think he's muted.
Yeah.
Okay, I'm unmuted now, yes?
Yes.
Hi, Kim. How are you doing?
Hi, Joel. Great to see you. Great talk.
Here. Thank you.
Thank you. So I also had a question about this first compound you described, the Cas-A inhibitor. Looks like that's further along in your development towards a drug, and it seems to have attractive properties. So you show that in the short infection model, you deliver 100 mg per kg, and then in the longer infection model, you deliver 5 mg per kg. 5 mg per kg is very impressive, but it's almost counterintuitive. Why do you go from 100 to 5 from a simple model to a difficult one?
Right. So it's a good question. So in the acute model, we actually did not titrate down. And so I don't actually, no, let's see. We did not titrate down. So Eric and Nurnberger, through the TB Alliance's support, looked at our compound in his acute model, which, as you may guess, is slightly different than the one that Veronique runs with David Perlin and Steve Park at Hackensack. And the dose response is roughly similar. So so I think that the minimum effective dose in Eric's acute model was close to 5 mg per kg. So I think that. So that's the dose response. I think that, certainly you're seeing a rather flat, dose response or not. And actually, maybe I don't show that data, but I can say it anyway, just to be, we don't gain a lot above 20 mg per kg. We haven't seen anything odd in terms of the PK. I don't know if it's something at the level of target engagement, a la, Pete Tong, but that's essentially the dose response.
Thanks.
Thanks, Kim.
There we go. Here's another question from another legend and another friend with a good sense of humor. Fred Ausubel asks, the quinoline compound sterilized the MRSA cultures in contrast to vancomycin. Does it kill the persister population?
That's a really good question. We honestly have not looked at that yet. As you probably could gather, we were and still are rather newbies, I guess, in the staph world, and so we relied on Jeff Boyd's lab when we jumped into the staph genetics. We did not actually do that yet. That's a really good experiment to try, and we're aware of that in other sort of programs that we've seen. So no, unfortunately, we have not done that yet. But we will definitely. I'll make a note. Thank you.
Great. Here's another legend and a friend. Ralph Isberg says it's not clear that NO is actually involved in killing in all cases. If NO is irrelevant for killing, and in some cases it may be, could you redesign the antimicrobial so another leaving group is added rather than and broaden the number of candidates?
Yeah, that's a really good question, and it's great to see Ralph or hear Ralph's question. Right, and so this was interesting to me because honestly, I feel like there's been mixed messages in the TB field, and that's not meant as a criticism at all. I think it's more maybe an evolution of thought. I think that when you look at the papers on protominid, beautiful papers from Cliff Berry's lab, I think it really gets across the point that the release of NO is tied to activity. I think that, especially those of you on the, on this seminar and know other bacteria much better than I do, at least what I've gathered is that NO is usually static. In bacteria static with lots of other bacteria. So my sense here, I think what Ralph is saying is right on in the sense that it may just be the metabolism of 2019 that releases NO that actually is what's cidal. And at best we've seen, let's see, we've done some work. We published a little bit in the 2020 paper where we tried to correlate NO release with activity, and we do see that correlation. But the thing that we have not done that Cliff did in BCG in his original paper was to use basically a trap for NO. I think it's CPTIO. I don't remember the actual chemical structure of it. I should. But that would obviously trap NO and prevent any toxicity that it potentially could give. But I think Ralph's right. I think that this is much more a question of what else is being engaged in here in terms of the biotransformation of the parent compound that's leading to cidality. It definitely appears to be F420 related, but I think there's still much more to be understood. So I completely agree with Ralph. I can't add more at this point, but I completely agree.
Let me just follow up a little bit. So on the ones where you looked at and showed biotransformation, did you focus on those for which biotransformation was essential for activity, or did you focus on those where biotransformation abrogated activity? What are you looking at there?
Right, that's a good question. So certainly in the case of 2019, we were obviously tried to see with mutations that conferred resistance, and so so that sort of shaped our investigations with 2019. I'm trying to think more broadly speaking. We've definitely seen mutations. Excuse me, I should back up. We've seen biotransformations that lead to a more active compound, So I think that we see biotransformations that run both ways, and also biotransformations that give you a compound that's maybe roughly the same MIC, but we don't yet know in terms of if that means a different mechanism. So that's really what shaped our investigations. And I apologize, I may have wandered away from answering your question. So if I could trouble you,
that was really right. And I'm wondering how knowledge about biotransformation might feed into your model and
right.
drive you towards some compound.
Thank you. Okay. Oh, so you're more talking about the prediction of active compounds with,
Yeah.
that's an excellent point. You know, at this level, we're really, the model has no insight in terms of biotransformations. We are trying to work on that now. We're just limited with data. We just don't have enough data to sort of learn from that. There are certainly programs that can take a small molecule structure and basically predict metabolites. That's obviously going to be biased by what it's learned from. So I'm trying to think, you know, I think that ideally, you know, you'd love to see basically, like, for example, a molecule that be toxic to the bacterium and that engenders a response where perhaps an essential metabolite is called upon to basically be involved in sort of the detoxification. You know, that could be one example. Or you could have a case where the bacteria tries to detoxify the molecule, and so doing, it actually creates something that's more toxic. So I think those are roundabout ways. I should get back to your question. So we have not yet been able to incorporate that in the models, but I completely agree that that's the more data we get about intrabacterial drug metabolism, the more able we should be able to build predictive models.
It'll be super interesting. Hannah, do we have some hands up there that you're
Not
seeing?
hands, but I see a related question. This is a good follow-up from Julia Kohler, who asks do you know the fate of these biotransformed compounds? Specifically, do bacteria release the biotransformed compounds, and do you have to look at their toxicity to the host?
Yeah, that's a really good question. So with TB right now to date, and I'd really be curious if any TB folks are on the call, we've been limited in our ability to basically inspect the media. And our general sense is that because, you know, you need surfactant to basically grow TB and not have it clump, that surfactant really kind of challenges your chromatography and your mass spec analysis. I don't think that's an absolute roadblock, but that's at least hindered us to date. And whether that's, you know, a good thing or not, I'm not sure. But, you know, I think we can potentially get over that. So we've not been able to assess so much how much these metabolites are released into the media, right? So that's one aspect that I think we need to understand more about and try to gain a mass balance in our system. And I think that, like, for example, in the 2019 story, you'll note that, you know, we have a hint that basically other metabolites are involved in terms of affecting fatty acid biosynthesis, because of, you know, we see this when we use the FGD1 transposon insertion, we also lose this FAST2 inhibition phenotype, but we have not been able to find any other metabolites. So this, you know, is a limitation with, you know, with using mass spec, is that, you know, you have to ask, is something there? And then you look at the mass spectrum and see if it's there, or do you have to be able to kind of sift through, you know, all the peaks and try to discern if something is, you know, is being produced from your compound. And obviously there are ways to do that with isotopic labeling. And so we are trying to pursue that. Finally, back to the question. Yeah, that's a really good point as far as toxicity to the host. And so I think we generally think that the metabolism within bacteria is been underappreciated. And I think it's being studied more and more. You know, I mentioned, you know, the Nathan lab, the Ree lab, Cliff Berry's lab. So I think that really will allow us to gain some insights into toxicity. So I think that the question is right on, that we really do need to be asking, what's the ultimate fate of drugs that were being used to treat bacterial infections? And, you know, besides just traditionally asking, okay, we have this drug, we're going to subject it to human liver microsomes, or we'll dose mice with it and see what the metabolites are in uninfected mice. I think we need to do that also in infected mice, and just also with bacterial culture, to try to understand what are the biotransformations we're getting because of the bug-drug interaction.
Great. I see a question from Dr. Sri Suk Nimit, and it gets to the informatics end. I think we're generally more comfortable with the microbiology and even the chemistry, but the informatics is newest to all of us, or at least certainly to me. And the question here, the specific question, is how do you filter against generally toxic compounds like membrane disruptors? But if you would maybe even broaden that a little bit in terms of how you inform the model.
Yeah, that's a great question. And so one way we do that is just experimentally. we will basically cast our net as broad as possible. So for example, in the staph model, we didn't consider toxicity to Vero cells, for example. And I neglected to mention, but before Jimmy Ray's mutants, he looked to see if the compound released NO. It does not. He looked to see if the compound depolarized the membrane. It does not. So we had that sort of information. It's worrying about something that just was a membrane disruptor. I think what I would say is that from our point of view, this is also an opportunity for predictive models. And so, we built a machine learning model for Vero cell cytotoxicity. I think that one can also turn to other data sets, right? So if you have where you've done a high throughput screen, for example, for molecules that disrupt the membrane, that's again data that one can learn from. And hopefully it came clear during my talk that one can build a model with a few hundred compounds. I think the key aspect is really going to be how diverse are those molecules, because you'd imagine the more diverse your training set is, the more your domain of applicability is in terms of being able to extend out to new compounds that are distal from your training set. So I think the question is right on. I think that I think you need to try to filter if possible computationally. But also, I think that it just, again, underscores those experimental studies that you need to do once you have a hit. And so besides looking for things like solubility, metabolic stability, you want to look at those things as far as membrane disruption, some general cytotoxicity to mammalian cells.
Great. Hannah, any other hands raised that we haven't gotten to?
I don't see any hands now, no. And I did answered more or less the question I was going to ask about building the model and have you encountered a strong lower limit? But it sounds like it's both the numbers and also the diversity of compounds that you're able to train it on. So that's really helpful. Oh, we do have one more from Kim here.
Oh, sure.
Yeah, if you could indulge me, Joel.
Always, Kim.
Thanks. I had a general question. There is this interesting phenomena that you can discover compounds that become drugs to hit TB. And people, including you, are doing that better than in all other fields of microbiology combined. And so one possibility is that people who work with TB are just smarter than the rest of us. And so that would be a trivial explanation.
Okay.
But I wonder, there's another possibility, and I wonder what your thought is about this, is that in TB you use combinations, and you only use combinations. So compounds with high frequency of resistance, like some that you showed us today, they are not banned from development. And so you now have access to all targets. And in creatures like staph or E. coli, where the expectation is single drugs, that collapses the universe of targets to a small handful.
Right. Yeah, that's a really good point, Kim. And I appreciate that very much, because I think, you could look, for example, at bedaquiline, right? And the issue with that relatively high frequency of resistance. And yet, as you know, probably as well or better than I, it's protected in a way in the clinical treatment with bedaquiline and linezolid. So I would say, yeah, that I think your explanation of that, and not the TB researchers being smarter, but the idea of combinations, I think that really, I think that's a big aspect of the relative success. Although, one thing I would say, and I maybe turn the question around to you or anyone else who's on the call, it's amazed me that, so for example, if you look at high throughput whole cell phenotypic screening against TB, we're typically very happy if we get a hit that has an MIC, let's say 1 to 10 micromolar. Whereas for malaria, and they've done just wonderful high throughput screening work, Elizabeth Winsler, David Fiddock, and many others. And they just seem to find, like nanomolar compounds, whole cell actives, like there's no tomorrow. And so let's see, and right, obviously there you're often looking at combination treatment, although not as many drugs, right? It's usually a two drug combination, if I remember correctly, for malaria clinically. So I'm not sure if that tells you something about, I think your frequency resistance in combination is a really good point. I don't know if that tells you something about compound accumulation, and right, I don't think we've learned that yet about malaria. I don't know if it has to do with metabolism.
Sure. What you're saying is that it's even easier in malaria, right? Where
it appears
you do not have a bacterial permeability barrier. MTB doesn't have a terrific permeability barrier like gram-negative bacteria, but it's certainly still better than in malaria.
Right.
That may be the difference.
Mm-hmm. Yeah, but I really do appreciate your point as far as the combination. And I guess I don't think, I'm trying to think about like clinical trials or stuff like that. I mean, I don't think they really, or the, I forget what the name of the program is, but I think they're always looking at least a 3-drug combination, right? I don't think you're ever looking clinically at 2 drugs. I may be wrong on that, but maybe that's, again, just it's automatically working to your advantage as long as there's not some sort of antagonism that you're dealing with TB, at least 3 drugs.
Yeah, thanks.
Okay, great. Thank you so much, Kim.
Great. Well, Joel, I can't thank you enough for helping us kick off the semester in style. That's going to be a tough act to follow for all the following months. We will have our monthly MSI seminar series, so keep watching your emails from Hannah for advice on that. But with that, I'll be happy to buy you a beer as soon as we're back to normal, and the next time our paths cross, which I hope is sooner rather than later. So thank you very much again. Joel, last words to you.
Oh, yes. No, thank you so much, Mike and Hannah, and Julia. It's really a pleasure, and everyone who has took the time. I hope you all stay safe. I look forward to get up to Boston. I miss Boston greatly. And a shameless plug, we're looking for multiple postdocs, computation, chemistry, or biology. So please, if anyone's interested, just drop me an email, give me a call. I'm really happy to chat with anyone. So.
Oh, fabulous.
Great. Thank you so much. Anything to add, Hannah, before we sign off?
No, ditto to everything that you said, Mike, and thanks so